Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Many species of lizards can drop their tails to distract a predator while they make their getaway, an ability known as tail autotomy. The animals then regrow their tails, but a regrown appendage is simply a cartilaginous tube. It lacks all the skeletal and neural structures of the original tail. Now, researchers from the University of Southern California have discovered a way to allow mourning geckos, Lepidodactylus lugubris, to successfully regrow perfect tails, says lead study author Thomas Lizito, a regenerative biologist at the institution's Keck School of Medicine. He says that he and his colleagues are particularly intrigued by the nerve growth that occurred, because it has implications for one day helping spinal cord injury patients to be able to form new nerves. Katharina Becker, a professor of neural development and regeneration at Dresden University of I think that is always very difficult to gauge. That said, she continues, the insights from this study can have, ultimately in 10, 15, 20 years, a huge effect on regenerative therapies, although they may not immediately be obvious. Lizito and his colleagues had previously shown that adult neural in a developing embryo, however, hedgehog is only produced by neural stem cells on the bottom, or ventral, side of the tail, freeing top, or dorsal, ones to develop into skeletal and nerve tissue. To mimic this embryonic state, the team extracted neural stem cells from the spinal cords of the gecko's tails and employed CRISPR-Cas9 editing to knock out a gene called SMO. Histology of a fully regenerated mourning gecko tail. Muscle is colored in white, cartilage in red, proliferating cells in green, and cell nuclei in blue. Ellie Tanaka, who researches vertebrate regeneration in salamanders at the Research Institute of Molecular Pathology in Vienna, describes the idea of using advanced gene editing techniques for regenerative studies, very powerful, and futuristic. She wonders if it would be better to perform the general. Another open question is what neuronal cell types make up the nerve networks observed in the regenerated tails, she notes. But for now, she says, she's impressed that such a straightforward genetic engineering was enough to endow a new tail with tissues it normally lacks. It's one minor change that has a systemic influence on the entire structure that is regenerated. That makes me really optimistic that you can improve regeneration in a structure that regenerates, but not perfectly, by a relatively minor intervention. Lizito is now trying to induce the geckos to regenerate their legs, which they normally don't. So far, his team has injected the tail-derived neural stem cells into amputated leg stumps and seen tails grow in these abnormal locations, he notes. We have lizards in the lab that we have just covered in tails everywhere. Rocky Tuan, a tissue engineer with a focus on regenerative medicine at the Chinese University of Hong Kong and Lizito's former advisor, notes that the choice of study organism here is important, as asexually reproducing Mo. The beauty of this is they're all clones, so you can do any transplantation that you want. Becker calls this bypass around triggering immunosuppression a benefit unique to this particular animal model. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.